So this is the ninth Cyclodextrin Masterclass that we initiated almost a year ago. And as I mentioned, we'll have a general overview today preparing for CPHI. So after very brief general introduction of cyclodextrins, we'll talk about how they are used in small molecule delivery, then in biologics, then other uses in pharma, and at the end, non-pharmaceutical applications. We'll close the topic. So, as you may know, cyclodextrins are made of starch. They are ring-shaped molecules, and the peculiarity of the cyclodextrin is that this cavity, the whole of the cyclodextrin, has a hydrophobic nature. So, it's an ideal environment for hydrophobic molecules to sit in and form a temporary inclusion complex, which is reversible. So, the cyclodextrin can release the active ingredient easily. And this phenomenon is exploited by a lot of different industries today. As I said, we'll focus on pharma, supplements, and cosmetics, but they are there in households, foods, textiles, agrochemistry, and a lot of other applications. While the dominant use of cyclodextrins is small molecule encapsulation and delivery, there are novel applications which are growing and emerging, and we'll also talk about these today, which are using them as active ingredients, formulating biologics, gene therapy, or monoclonal antibodies, or uses in biotechnology. So how this whole process works, we have an empty cyclodextrin with this hydrophobic cavity, and we have a hydrophobic API or an API that has hydrophobic moieties like aromatic groups or something like that. And in a thermodynamic process, an inclusion complex can be formed. There is no covalent bond created. It's just a temporary complex. Technically, the active ingredient is jumping in and out of the cyclodextrin cavity constantly. Why is it good for us? What do we observe when this complex is formed? We observe an increase in bioavailability, so we can reduce the dose of active ingredients. We can mask the taste or the smell of active ingredients. We can often decrease interactions with other drugs or irritation and side effects. What do we see as an increase? The solubility is the most typical among all, but also the dissolution rate is quicker with cyclodextrins, so fast onset applications are easy to make. The chemical or physical stability of the encapsulated ingredient can grow substantially, the wetting, and of course, according to all these, the bioavailability. So, how do we deliver small molecules with cyclodextrins and what kind of products are there? Since I'd like to make this a bit interactive, there are a few questions embedded into the presentation. So you'll see that cyclodextrins are used in general very widely in the pharmaceutical industry. There is one administration route where there are no products on the market. What do you think it is? Very quickly, guesses in the chat box. If you have any guess. If not, I'll tell you. Okay, so we get to that. It's the pulmonary application, which only have products in clinical phases, not on the market yet. As I said, cyclodextrins are used very widely in the pharmaceutical industry. First of all, you should know that when talking about cyclodextrins, we talk about different types of derivatives. So as you can see on the picture, there are 7 different cyclodextrins that are approved for pharmaceutical purposes. You can also see from this table that depending on the administration route you pick, you cannot use all the 7 cyclodextrins necessarily. So if you go with oral, you have a certain limitation. If you go with topical, you have another set of limitations, which is from one point of view due to the availability of existing safety data, and from the other point of view is because some cyclodextrins are not safe in certain administration routes. For instance, I don't know, the beta cyclodextrin is completely okay orally, it's even on the GRAS list, but if you administer parenterally, it would cause severe side effects. But again, seven cyclodextrins and tackling all the administration route pulmonary also soon. When you try to make a cyclodextrin complex, luckily you have a very wide variety of formulation technologies to pick from. Typically, it's a fairly simple process compatible with any kind of equipment. So you can make this complex in liquid phase, uh, like creating a common solution or a common suspension and isolating the complex. But you can do also it in a semi-solid phase with an extrusion or granulation type of technology, and even in solid phase with the commiling. Of course, the efficiency of the complex formation is not identical among the different techniques, but the possibility is there depending on your capabilities and the characteristics of the active ingredient. So, what does it looks like if you have a good complex or not good complex? Because in many cases, people think that just mixing the active ingredient with the cyclodextrin would mean that the complex is formed. And this is what you see here in this short video. So, the first picture doesn't contain any cyclodextrin. In the middle, you see a mixture, just a physical mixture of the two, and a true complex is seen in the last one. So you can see the huge difference. Cyclodextrin can still improve the wettability of the compound, but the difference is rather dramatic. And of course, if we go into analytics, it would be even more substantial. Just to show you a few examples about the power of cyclodextrins, first it's about solubilization. How much media do we need to solubilize something? I brought up the example of alfexalone. Here you can see that without cyclodextrin, you would need about 2 liters of media to dissolve 10 milligram, and with cyclodextrin, sub-milliliter volume would be sufficient. Bioavailability increase can be really dramatic, and some products really take advantage of that application, like a recent glucagon formulation called Baqsimi, where the bioavailability was improved sevenfold with the use of the cyclodextrin. And increasing the stability can also be a very straightforward use. So you can see from 2 months to 2 years, stability increases completely feasible. When it comes to stability, we're not just necessarily talking about physical or chemical stability. A proper cyclodextrin complex can provide a very wide range of protection. So it can protect against light, heat, oxidation, as you can see, pH, or even moisture. Among these five, the moisture is the most tricky, as usually the complex formation happens in an aqueous media, but it can also be done without it. So even compounds that are prone to hydrolysis can be encapsulated into cyclodextrins. Enhancing permeation as an application is a rather peculiar one, because we mentioned that cyclodextrins can improve bioavailability, which is true. Cyclodextrins usually do that via improving the solubility. So it's not a direct permeation enhancer in most cases, but something that makes more active ingredient available for permeation. They can also enhance the permeation itself, but for that, the cyclodextrin has to interact with the membrane surface. So cyclodextrins can, as shown here, encapsulate membrane components, so like cholesterol or fatty acids in the membrane, and improve the fluidity of the membrane, helping the compounds to get through. This is possible with cyclodextrins, but for this, usually you need a high local concentration of cyclodextrins. So you need a special formulation, something mucoadhesive, for instance, or you need an administration route where the dose of the cyclodextrin on the surface of the permeation area is high. So topical, nasal spray, stuff like that. Like the Baqsimi that I showed you is a nasal spray. So in these cases, permeation enhancing effect is real. I'm just bringing a few examples from around the world, marketed products where cyclodextrins are used not for solubilizing. So, as mentioned, taste masking, quite typical. Ensuring content uniformity for highly potent APIs, it can be a possibility. Uh, as I mentioned, the release of the cyclodextrin cavity, the active ingredients release is usually very fast. So developing compounds that are or dispersible or has or need a very fast onset is a very ideal way to use cyclodextrins. Chemical stabilization and life cycle management as well is a good possibility of using cyclodextrins. Just to show you, because I haven't talked about this, what kind of dosage forms cyclodextrins are used in. We talked about administration routes, but not dosage forms. They're very compatible with all kinds of dosage forms. Most products are oral tablets and capsules. Parenteral liquids or lyophilized parenteral products are also very common, so are eye drops. But as you can see here, you can make gels, you can make suppositories, you can make wipes, chewing tablets, chewing gums. Technically, you can incorporate cyclodextrins into more or less anything. So, the second part is about biologics. This field is much more novel than the small molecule delivery. So the small molecule delivery dates back to the 1970s. Um, and for biologic delivery, we are talking about a history of about 15 years only. And there are not too many products, as you will see on the market, but there are a lot with what are in development. So what do you think is the primary use? Again, a question in the chat box, please be proactive. What do you think is the primary use of cyclodextrins today in biologics? I already explained how they are used in small molecules. So what is your feeling? Why would we use them in biologics, in protein formulations and so on? Maybe the chat is dead. That must be a Zoom issue. I cannot imagine that no one has a guess. Okay. Anyhow, now, okay, good. We have stability, protecting from proteases. Okay. Uh, yeah, the stability enhancing effect is two. Protecting from proteases is not true. Okay, let's get to the bottom of this. Physical stabilization is the typical use. So cyclodextrins can prevent the so what are the let's start from the beginning. What are the issues with the proteins? There are quite a few. So proteins can aggregate. In some cases, although not very common, but proteins do not have proper solubility. Since we are usually using very high concentration solutions, they can get very viscous, absorb on the surface of the packaging, and also degrade. And cyclodextrins can help with all of that, depending on the circumstances. Um, Moreover, when we compare, so the physical stabilization is the dominant here. Prevention of aggregation is the most typical application of cyclodextrins for protein formulations. You were right about that one. Um, of course, here we have a direct comparison because this is a problem that can be solved with other components like polysorbates. So cyclodextrin is technically competing with those. What is an interesting study from Rocket shows, however, that you don't necessarily have to think about it as a competition. You can also think about it as a synergy because the mechanism of action for cyclodextrins for protein stabilization is different than that of the surfactants. And as you can see here, uh, we could see more or less a similar stabilizing effect when we used hydroxypropyl beta cyclodextrin or when we used the polysorbate. But if we use the two combined, you can see that the effect dramatically increases. And that's because the cyclodextrin has a two technically a double mechanism of action of protecting the proteins. So there is one on a molecular interaction level. Of course, the cyclodextrins are capable of interacting with amino acids on the proteins. The hydrophobic amino acids can be encapsulated in the cyclodextrin, and in this case, the cyclodextrin provides kind of an outer surface protection, but there is also a surface activity which is similar to polysorbates. So it doesn't necessarily need encapsulation, but via hydrogen bonding, it can provide a surface covering effect. And the combination of these two can have a synergistic effect with other stabilizers. How does the viscosity reduction work? Okay, it's not very correct to say it's a viscosity reduction. It's a prevention of viscosity increase. So the cyclodextrin also, via preventing the proteins from aggregating, keep them separate, which leads to technically a viscosity reduction, but technically it's a prevention of viscosity increase. And a similar effect happens with the prevention of absorption. The proteins that have their surface covered with cyclodextrins cannot really interact with the wall, which also has some cyclodextrins in it. You should know that when it comes to formulations, and I haven't mentioned this before, irrespective of small molecules or biologics, usually the cyclodextrins are in excess in the solution, which means that there are cyclodextrins which actively interact with the protein or the small molecule, and there are always empty cyclodextrins in the solution, as you can see, which can be there on the capillary wall or the container wall and so on. As I mentioned, there are not too many commercial examples of cyclodextrins in biologics. One is a fairly recent one, which is J&J's COVID vaccine that contains hydroxypropyl beta cyclodextrin. Um, the exact purpose of containing this cyclodextrin is not specified, so you have to read between the lines. But there is a two-way effect. Of course, there is also a stabilization effect here. But the cyclodextrin is clearly also used as a cryoprotectant, so to improve the stability in the freeze and thaw process. Cyclodextrins are known to have that activity for quite a long time, especially from veterinary uses. And this was also one of the features that was exploited by J&J in their vaccine. When it comes strictly to protein stabilization or protein formulation, the closest to this application or the closest products are Kyprolis, which is of course not a protein. I'm not saying it is. It's an oligopeptide where the purpose of using cyclodextrin is still solubilizing. So this is, I understand, it's closer to a small molecule. And the glucagon of Baqsimi, which we mentioned, which is a nasal spray. So here the stabilization is not necessarily the effect again, but at least it's a protein. Um, just as an interesting feature, there are some approaches of using cyclodextrins in antibody drug conjugates, where the cyclodextrin would um, help deliver a payload or yeah, technically the cyclodextrin's role would be to use them instead of PEGs and other linkers in delivering a payload. It's nothing close to human applications, but it's certainly a possibility to create something like that. The third part, oh no, fourth part of the lecture is other pharma uses. So in this case, cyclodextrins are not excipients as they were discussed so far. So first we'll talk about cyclodextrins as active ingredients. This is a very old story. As you can see, the first paper here was published more than 40 years ago. Jesus, okay, so it's older than me. When cyclodextrins was used as an active ingredient, this particular cyclodextrin, which is currently not in pharmaceutical use, was administered with the specific aim of removing something from the body. So it was a hypervitaminosis, a very severe hypervitaminosis, and this was proposed as a solution to save a kid in this treatment, and it worked very well. So technically, the concept is that you have something accumulated in your body, you introduce the cyclodextrin, which finds it, encapsulates it, and removes it from the body. And in this case, it worked beautifully. And then came a long silence until we arrived to Sugammadex, which probably you all know. It's marketed under the trade name Bridium. It was approved in the EU in 2008 and in the US in 2015. Um, so this product has a very similar concept to the hypervitaminosis, only it's not an intoxication we're talking about, but the deliberate removal of a xenobiotic. So during surgery, there is um, a neuromuscular blocking agent called rocuronium introduced to patients, and Sugammadex was developed and via systematic medicinal chemistry designed to be very specific, very selective for rocuronium, so that when we administer Sugammadex, it goes through the blood vessels, it finds all the rocuronium that is bound to receptors, it captures the rocuronium, removes from the receptor, and the bonding here is so strong that it's never released again. So it's a covalent strength of bonding we are talking about. When the complex is formed, it never dissociates again. There are such cyclodextrin complexes existing. So it's a selective removal of something. This is a blockbuster drug, ladies and gentlemen. So um, it was also successful when it was just in the EU market, but after approval in the US, you can see that it developed very nicely. Patents are mostly expired, so now we are not just talking about MSD, but a lot of generic players. Like I think there are around 30 plus companies who has patents on Sugammadex manufacturing and so on. So it's something that you'll probably see a lot at CPHI. This is the only approved cyclodextrin active ingredient currently. But okay, before that. So um, before we go into more what's going on and what's happening in the cyclodextrin as active ingredient field, what do you think? How many companies have cyclodextrin as an active ingredient asset? So how many companies develop cyclodextrin-based active ingredients today globally? 10. Okay. Let's see a few other guesses. 100. Very ambitious. So the truth lies between the two. It's over 20, technically. So I will not go into detail who is doing what, but all these companies are pharma companies, and they all have inventions or developments that involve cyclodextrins as active ingredients. To give you an overview about this one, just on a very high level, we are talking about two different concepts. So two substantially different mechanism of actions. So the first is the scavenger team. Of cyclodextrins. So here the mechanism of action is identical to Sugammadex. So you have a cyclodextrin and the goal is that you administer an empty cyclodextrin. Okay, in both cases it will be empty. An empty cyclodextrin that has an internal small molecule target which it has to remove from the body. So what is the small molecule target? Because as you'll see, it will lead to a lot of different applications. As you have seen, the hypervitaminosis and the Sugammadex are very different. Stargardt disease, it's accumulated lipofuscin. There is a clinical study where it's low molecular weight heparin. So to stop the effect of those molecules. There are other compounds that are developed for the exact same application as Sugammadex, removing a toxic version of cholesterol. In fentanyl overdose, removing the fentanyl, or even organophosphorus intoxication. So the target can be very different because cyclodextrin chemistry is very versatile. You can really tune the cyclodextrin so that it's very selective for your compound. It can be anything, more or less the application. Of course, it has to be something that can be encapsulated into cyclodextrins, but you have a lot of options chemically. The other is the cholesterol leak. So in all the other diseases, the mechanism of action is a bit blurry, but it was it was discovered around 15 years ago, let's put it like that, maybe closer to 20, that when we administer this particular cyclodextrin, which is an excipient, a parenteral excipient, the hydroxypropyl beta cyclodextrin in high dose, it has beneficial effects on cholesterol homeostasis, especially when cholesterol is accumulating within the body. The primary application was Niemann-Pick C, which is a rare disease where within the lysosomes cholesterol accumulates, and it was discovered that if you administer the cyclodextrin, it can help to remove the cholesterol. But how? Okay, how is debated because there are different theories. And in my opinion, my personal opinion, none of them has been unambiguously proven. But what is proven is that there is a positive effect on cholesterol homeostasis. So all the diseases that has a defect with cholesterol homeostasis, which are a huge number, by the way, were tested and were proven to have a positive response for cyclodextrin treatment. So I'm just listing here some of these so that you can imagine Alzheimer's, Parkinson's, atherosclerosis, different types of venom poisoning, FSGS where cholesterol accumulates in the kidneys, certain types of cancers, gout, lymphedema, certain eye diseases. So there is a lot of pathologies where cholesterol accumulation or impairment in cholesterol homeostasis is an issue, and this cyclodextrin seem to help in those diseases. What is beyond this cyclodextrin? So uh, we have seen companies who approach the issue not by using this particular cyclodextrin, but designing something that's for cholesterol, and what can we do? But here the big question is, okay, how is the cyclodextrin working? Is it direct encapsulation of the cholesterol? Is it a receptor binding? Is it triggering a cascade? Is it a sync type of effect? So there are a lot of options for this one. Two things to consider just to summarize this active ingredient story. Um, so there are the scavengers which have a clear target, which are really custom-made cyclodextrins for the target, and the aim is to remove something. The question here is that, okay, we can remove something which is a xenobiotic, which comes from outside. Can we remove something which is already endogenous? Can we design a cyclodextrin just to remove something like cholesterol, which we need? But okay, if it accumulates, it's causing problems. So can we remove just part of it or selectively remove some certain cases? These are for the future to ask in this case or to answer in this case. For the cholesterol modulators, as I said, in my opinion, the mechanism of action is not really clear. Dominantly, most of the companies use repurposed CDs. So here we're not talking about custom-made compounds, but compounds from the shelf, but they all influence cholesterol homeostasis somehow. So the question here is that interestingly, none of these companies have created or tested a cyclodextrin, which is a very good cholesterol targeting cyclodextrin. Interesting question is why it was never tried if this is the exact clear target compound. Another question that I haven't really mentioned so far. So when it comes to cyclodextrins chemistry-wise, we can talk about so-called single isomer cyclodextrins, which are very well-defined compounds, like most active ingredients. There is a chemical structure that defines the compounds. You know what the impurities are, you know what the metabolites are, you know what the degradation products are. And when in the scavenger team, most of the compounds are like this. Of course, to make such a cyclodextrin is usually fairly expensive. You need several step synthesis, and technically it's like developing any other new active ingredient. But you'll have a good understanding on what's happening with the cyclodextrin. Of course, we should know how cyclodextrin work if we want to design something like that. The other application, as I said, approved pharmaceutical excipients. The approved excipient cyclodextrins are not single compounds, usually. Okay. Uh, uh, um, Not talking about alpha, beta, and gamma cyclodextrin, but sulfabutyl, hydroxypropyl. These are mixtures of thousands of compounds. So you don't really know what is working within them or what is not working. If they degrade, you'll have no idea what are the degradation products because you'll have millions of degradation products. You'll have very limited idea of the pharmacokinetics. If it's causing any side effects, you will not know which component is causing the side effect. But it's something repurposed, it's something with a very established safety profile. So from a regulatory perspective, it's a lower hanging fruit. The question here is that so far we have said that these are, okay, let's not say inert, but functional excipients. So how do you make a functional excipient, or how do you transfer a functional excipient to an API from one day to the other? And what would that mean from a regulatory perspective? So as you can see, this application of cyclodextrins has some dilemmas. Okay, that's all about cyclodextrins as active ingredients. There are a few other uses still within pharma, but not active ingredient and also not excipient. So cyclodextrins can be used as vaccine adjuvants. It's also a long story. It started with Pfizer and sulfolipocyclodextrin, which is a very unique cyclodextrin. By the way, you cannot source it commercially, but it was uh, a component specifically as a vaccine adjuvant in certain veterinary vaccines. There are more, but I'm just bringing this to vaccine, which led to an interesting question. Can other cyclodextrins be used as vaccine adjuvants in vaccine formulations? And as it turned out, they can. So now the pioneer that there are again veterinary formulations that has cyclodextrins as active ingredients on the market. But in humans, the most advanced is Daiichi Sankyo's development. It's an intranasal flu vaccine where the hydroxypropyl beta cyclodextrin, the very same compound that was used as a parenteral excipient and later as an active ingredient, is now used as an adjuvant. It's unbelievable for me. But now it was proven that T-cell related immune response and local immune response was significantly boosted by the application of hydroxypropyl beta cyclodextrin. For me, the question is, okay, so how is it boosting T-cell related immune response? And if it's boosting T-cell related immune response, as shown, again, this cyclodextrin was on the shelf. Is it the best cyclodextrin to be used as vaccine adjuvant? Probably not. So here there is a lot of hows to be answers, and I really strongly believe that cyclodextrins, certain cyclodextrins, not necessarily this one, will emerge as a new class of vaccine adjuvants, a safe and potent type in the future. Vaccine production, completely different topic and just bringing one slide, but using cyclodextrins to improve the yield, so the productivity of certain biotechnological processes is very common. It can be the synthesis of certain active ingredients where bacteria are used, or it can be the synthesis of a toxoid, like in this case, the pertussis toxoid. The mechanism of actions differ, but cyclodextrins are there on the market for such applications. So in this particular example, when the toxoid is produced, there are two compounds, oleic acid and palmitic acid, created in the process as byproducts which inhibit the process itself. So as the production goes, these accumulate and then slowly shut down the whole procedure. unless you add a pinch of cyclodextrin into it, specifically this one called dimeb. It's the okay, it doesn't matter what is it chemically. The main point is that when you add cyclodextrins, these are hydrophobic compounds, of course, and cyclodextrins can encapsulate these compounds in the solution. And when they encapsulate it, they mask the presence of their compounds. That's cyclodextrins do. That's how they mask the taste. That's how they mask the smell. That's how they improve solubility. It covers the compound as if it was not there on its own. It's not existing on its own. And if it's not existing on its own, it cannot inhibit the process. So the productivity was improved 100-fold by adding 1% of the cyclodextrin into the solution. This is still how the toxide is made as of today. Finally, just very briefly, because this is still in preclinical stage, but cyclodextrin has the possibility of delivering genes as well. First, it was used as a combination product. So technically, cyclodextrins were combined with other gene delivery systems like lipid nanoparticles and so on to improve their performance. But nowadays, we're talking about standalone cyclodextrin delivery systems. We can make cationic cyclodextrin polymers like polyimines, and there are a few papers showing that they can deliver genes into cells. Polyrotexane, which is like a cyclodextrin necklace, can be used this way. Our particular focus at CarboHyde is amphiphilic cyclodextrins that can form a micelle-like structure, a nanoparticle very similar to lipid nanoparticles, but the building block is not a lipid, but a cyclodextrin here. We have data showing that we can deliver siRNA, mRNA, plasmid, you name it, antisense with very different cargoes into cells very efficiently. Or it can be like supramolecular assembly where cyclodextrin monomers can assemble and cover the oligonucleotides. So there are different approaches. Again, very promising field for the future. Very briefly, I'm not going into all the details of nutraceutical and cosmetic applications, just showing you a few peculiarities, some interesting stuff. Obviously, these industries use typically the cyclodextrins for the very same um, basic concepts as the pharma industry does, just the active ingredients differ. So it's not an active ingredient, okay, not a pharmaceutic drug we put into the cyclodextrin cavity, but coenzyme Q10 we put, and we see that the bioavailability is almost 20-fold with the complex. Or we have an essential oil, we can make a powder out of it with the help of cyclodextrin. Or we want to improve the solubility or an absorption of curcumin, you put it into a cyclodextrin and bam, you have a huge improvement. So again, bioavailability, solubility, stability improvement are the key applications. But okay, let me tell you something interesting. Alpha cyclodextrin. It's a very small cyclodextrin, typically for fatty acids, lipids, and it has a lot of uses mainly in the food industry, not so much in the pharma. There are a lot of clinical trials, so controlled human clinical trials when naked alpha cyclodextrin were administered to patients. So what is the sense of doing so? These basic cyclodextrins are used as dietary fibers technically. So they can be fermented by the gut bacteria. They can have a positive effect on the microbiome. They're safe, they're GRAS listed, and they can encapsulate fats of food that you eat. And it was shown that technically eating empty alpha cyclodextrin can have a very positive effect on both your blood sugar level and your blood lipid levels. And again, we're talking about clinical studies. So technically, alpha-cyclodextrin as a supplement on its own without any active ingredient exists, and it's a very nice supplementation for weight control and also a very cheap ingredient, by the way. The topic of creating cyclodextrin active ingredient conjugates has been around in the pharmaceutical industry for a long time. So technically a product, creating a product with a product pro-drug with a cyclodextrin. It's feasible. But it's a new chemical entity, so it was always debated, and early phases of clinics, some compounds reached, but nothing serious. The cosmetic industry, however, did make this leap. So formulating retinol and retinol with cyclodextrins has been known in the cosmetic industry. So was that cyclodextrin can be used in microneedle technologies. You can actually create a microneedle itself simply from the cyclodextrin complex, the gel-like structure. Um, but there is a recent invention which combined the two. So they created cyclodextrin microneedles and delivered retinol not as a cyclodextrin complex, but as a cyclodextrin active ingredient conjugate. And again, despite the cosmetic application, clinical data was generated to show the improvement in the functional properties, so the bioavailability or the wrinkle improvement, uh, as well as in the product characteristics. So it's a nice story, something that seems to be trivial for a pharmaceutical use could reach its potential in the cosmetic industry. Similarly, cosmetic industry metal organic frameworks, very actual topic. Just yesterday, Nobel Prize was awarded for it. So you can make cyclodextrin-based metal organic frameworks too. It looks like this one. And interestingly, I looked it up, and one of the laureates has cyclodextrin based paper. So they also have metal organic frameworks created with cyclodextrin. And even more interestingly, the paper was co-authored with the former Nobel laureate, Sir Fraser Stoddart. So two Nobel laureates worked on cyclodextrin based metal organic frameworks. Here, the concept is, of course, again, to deliver active ingredient. But what they did is, okay, let's not use this in pharma, let's use it in cosmetics and create a luxury brand where we show that such a delivery system can have an improved skin penetration. The same would work in pharma, of course, but more complicated regulatory-wise. You can have a very high load of the active ingredient, and boom, you have a luxury cosmetic brand. So what I really wanted to tell you today is that when you look at cyclodextrins, do not just look at them as solubilizers, because that's the past and maybe part of today's reality, but certainly not the future of the cyclodextrins, because small molecule formulation will be there, but there will be proteins, there will be vaccines, there will be genes, and a lot of advanced delivery technologies, both in the pharma field and close to the pharma field. So that's what I wanted to tell you. I hope I could share something new if you are going to CPHI or not. And I'm really happy to answer all your questions. I'll stop the screen share and I look into the chat box to see what do we have there. Uh, but if not yet, you can start with the questions. How they judge selectivity in blood if there is a lot of material that can fit the cyclodextrin cavity. Yes, that's very true, Mohammed. So you got the point exactly right. When we administer cyclodextrin into the bloodstream, a lot of molecules compete for the cyclodextrin. So in order to have the scavenger effect, I guess that you are getting there, and that's what you're asking about. You need an extremely high selectivity. So technically, before injecting, you can use human blood uh, as a media to show what it encapsulates and what it does not encapsulate, or even more so to see if the cyclodextrin, if an active ingredient encapsulated in the cyclodextrin is released. Typically, when we did such experiments, we were using ex vivo models, and yeah, technically, you have to use real human samples for the testing. The complex itself, you can. Uh, uh, you can analyze, as the cyclodextrin analysis is usually rather different from the typical analysis of small molecules. But it's a very critical question, especially when it comes to topic like, if you remember, I mentioned that one of the companies is working on removing a toxic metabolite of cholesterol. So when you try to remove something like rocuronium, which is at least a xenobiotic, so it's not something very highly similar to endogenous human compounds, you can imagine how difficult it is chemically to distinguish between cholesterol and 7-ketocholesterol, where the difference is just one oxygen between the two big compounds, and you have to be very selective. But synthetically, it's possible. So in this particular case, what Cyclarity did, I think the selectivity for the target compound, 7-ketocholesterol, is, I think, 1,000-fold higher than for cholesterol. So technically, you're not removing any cholesterol, just the 7-ketocholesterol. So yeah, it's possible, but you have to be very careful with that. And also, of course, if you administer a lot of empty cyclodextrins, those empty cyclodextrins will have targets in the human body. Let's go to the example of Sugammadex. Okay, it was designed for rocuronium, true, and it was very selective for rocuronium. But still, when you administer Sugammadex, you have a lot of empty Sugammadex circulating in the system, which will encapsulate other compounds. So it was discovered that, although not very selective for it, um, for instance, entry contraceptives, female entry contraceptives, are encapsulated by Sugammadex, and their effect is substantially reduced by Sugammadex. So technically, it's in the patient leaflet that I don't know if you are on the pills. Intercourse is not suggested for a few days because the effect will be reduced because of Sugammadex. So it can happen as a side effect. Other questions? I just scroll. So yeah, Dr. Anton, not you, Mahmoud Beish. Okay, maybe I just butchered your name. Sorry. So you mentioned with the question that cyclodextrins can protect proteins from proteases. And I haven't really mentioned why. I said that it's not correct, but haven't really explained why it's not working. Why it's not working is because of the usually fairly weak interaction between cyclodextrin and the guest molecule, should it be a small molecular protein. So yeah, we have talked about Sugammadex and this very strong binding, but it's very rare, and you need to tailor the cyclodextrin so that this binding is this strong. Reality is that usually the binding of the cyclodextrin to the gas molecule is either weak or at best moderate, which means that the cyclodextrin releases the active ingredient very easily, typically upon dilution. So what happens? We have a formulation, a tablet or a liquid concentrated in a small vial, syringe, whatever. So when you swallow the tablet and it dissolves in 2 liters of gastric media, or when you administer the injection and it gets diluted with 5 liters of blood, the dilution is huge. And as Mohammed pointed out, there is competition. And what will happen is that the cyclodextrin will release the active ingredient almost immediately. So if you want the cyclodextrin to protect your compound or your protein from something that's within the body, your job is much more difficult because for that you need a strong interaction, which usually doesn't happen. So that's why we have tried, but it's not really working against proteases because cyclodextrins are not there anymore to protect the the protein. If the interaction can be maintained, it is possible, but this is usually not the case. Yeah, I'm happy if it was understandable. Any other questions? Well, if you, yeah, if you don't have, or I don't know, if you would not like to ask, you can always find us on social media or just write to CarboHyde. We are really happy to answer any questions related to cyclodextrins. And yeah, as you said, Silke, yeah, see you at CPHI. And yeah, you all have a nice day. Okay, take care. Have a good one. Bye bye.